Welcome everyone. I'm excited to kick off today's presentation with our first slide, Biosynthesis of Fatty Acid. In this session, we'll explore how fatty acids are built within the body, covering key pathways and enzymes involved. We'll also look at how this process impacts health and disease. So let's dive in and uncover the fascinating world of fatty acid biosynthesis together. Let's start with the basics of fatty acid synthesis. The initial input is acetyl-CoA, which is a 2-carbon compound. This molecule undergoes a process called carboxylation, where a carbon dioxide molecule is added, converting it into malonyl-CoA, a 3-carbon compound. This step is crucial because it prepares the building block needed for fatty acid chain elongation. Now, this carboxylation process depends on ATP, meaning it requires energy input. This energy investment is essential for driving the reaction forward. Later on, during the actual condensation step, where the fatty acid chain grows, the CO2 that was added earlier is actually released. This decarboxylation helps to make the process energetically favorable. Interestingly, the decarboxylation is spontaneous, which means it naturally drives the condensation reaction without needing additional energy. This entire sequence is a beautiful example of how energy input and chemical reactions are tightly coordinated in fatty acid synthesis, linking back to the broader theme of how our body efficiently produces lipids from simple molecules. Acetyl-CoA carboxylase is responsible for catalyzing a crucial 2-step reaction in fatty acid synthesis. In this process, acetyl-CoA is carboxylated to form malonyl-CoA, which is a key building block for fatty acids. Now, as with many other carboxylation reactions, the enzyme uses a biotin prosthetic group, which acts as a carrier for the carbon dioxide. The first step, which is ATP-dependent, involves the carboxylation of the biotin at a specific active site. Think of this as attaching a carbon dioxide molecule to biotin, activating it for the next step. Then, at a second active site, the enzyme transfers that activated carboxyl group from biotin to acetyl-CoA, producing malonyl-CoA. This 2-step process highlights the enzyme's ability to efficiently link energy-dependent activation with the transfer of the carboxyl group, which is essential for fatty acid biosynthesis. Throughout this cycle, the enzyme's structure and function are perfectly coordinated to enable these reactions to occur smoothly, supporting the overall theme of metabolic regulation and energy use. The overall reaction we're looking at here is spontaneous, meaning it occurs naturally without needing additional energy input. And to summarize it simply, it involves bicarbonate, or HCO3, reacting with ATP and acetyl-CoA. This combination results in the formation of ADP, inorganic phosphate, PI, and malonyl-CoA. What's really important to highlight is that this reaction is a key step in the process of fatty acid synthesis. It essentially sets the stage for the building blocks needed to create fatty acids. The fact that it's spontaneous indicates that the reaction is energetically favorable, which helps drive the entire biosynthesis process forward. Connecting this to the bigger picture, understanding this reaction helps us appreciate how cells efficiently regulate fatty acid production, ensuring they have the necessary components for energy storage and membrane formation. Let's take a closer look at how biotin is attached to the enzyme. It forms a very specific bond, called an amide bond, which occurs between the terminal carboxyl group of the biotin side chain and the E-amino group of a lysine residue on the enzyme. This connection is crucial because it anchors biotin firmly in place. Now, think of the combined biotin and lysine side chains as a long, flexible arm. This flexibility is key because it allows the biotin ring to move or translocate between 2 different active sites on the enzyme. Essentially, this movement facilitates the enzyme's ability to carry out its function efficiently. This dynamic aspect, how biotin can shift between sites, is a fundamental part of how biotin-dependent enzymes work. It highlights the elegant way in which molecular structure and flexibility work together to enable complex biological processes. Understanding this connection helps us appreciate the precise mechanisms that underlie enzyme activity and regulation. Let's take a closer look at acetyl-CoA carboxylase, or ACC for short. This enzyme plays a critical role in fatty acid synthesis because it catalyzes the conversion of acetyl-CoA into malonyl-CoA. In fact, this step is considered the committed step, meaning it's essentially the point of no return in the pathway. Once it happens, fatty acid synthesis is well underway. Now, in mammals, regulating this enzyme is particularly important because fatty acid synthesis needs to be tightly controlled based on the body's needs. So, the enzyme's activity is regulated through two main mechanisms: phosphorylation and allosteric control by local metabolites. Phosphorylation involves adding a phosphate group to the enzyme, which can either activate or inhibit it depending on the context. This process allows the cell to quickly respond to hormonal signals and other regulatory cues. Allosteric control, on the other hand, involves metabolites that bind to the enzyme at sites other than the active site, inducing conformational changes that modulate its activity. These local metabolites act as signals, telling the enzyme whether to ramp up or slow down fatty acid synthesis. Connecting this to the overall theme, these regulatory mechanisms ensure that fatty acid synthesis is carefully balanced with the cell's energy status and metabolic needs, which is vital for maintaining overall metabolic health. Now, let's look at how conformational changes are associated with regulation. In its active form, acetyl-CoA carboxylase tends to associate into these multimeric filamentous complexes. This assembly is crucial because it enhances the enzyme's activity, allowing it to efficiently catalyze the conversion process. On the other hand, when the enzyme shifts to a non-active conformation, it dissociates into individual units, or monomers, which we call protomers. This dissociation effectively reduces its activity, serving as a regulatory mechanism. So the key point here is that the enzyme's activity is tightly controlled by its structural state, whether it's assembled into these filaments or broken down into monomers. This dynamic switching is essential for maintaining metabolic balance, connecting directly to how cells regulate fatty acid synthesis in response to their needs. So AMP functions as an energy sensor and a regulator of our metabolism. When the cell's ATP production isn't keeping pace with its energy demands, you'll see a higher proportion of adenine nucleotides in the form of AMP. This shift signals that the cell is low on energy. And what does AMP do in response? It promotes catabolic pathways. Those are the processes that generate ATP, helping the cell restore its energy balance. At the same time, AMP inhibits energy-consuming synthetic pathways, essentially putting a brake on processes that aren't immediately necessary when energy is scarce. A key example of this regulation is in fatty acid metabolism. AMP controls fatty acid synthesis and breakdown by regulating the availability of malonyl-CoA, which is a crucial molecule in these pathways. Overall, AMP acts as a vital checkpoint, ensuring that when energy is low, the cell prioritizes generating ATP and conserves resources. It's a perfect example of how our cells dynamically adapt to maintain energy homeostasis, which ties into the bigger picture of metabolic regulation we're exploring today. Let's break down how AMP-activated kinase, or AMPK, plays a crucial role here. When energy levels in the cell are low, AMPK gets activated and catalyzes the phosphorylation of acetyl-CoA carboxylase. Now, this phosphorylation effectively inhibits the activity of this enzyme, which normally promotes the production of malonyl-CoA. Since malonyl-CoA is a key substrate for fatty acid synthesis, its decrease means that fatty acid synthesis is significantly diminished. This is an important point because it shows how the cell shifts from building fatty acids to utilizing energy stores during times of low energy. And here's the interesting part. This reduction in malonyl-CoA doesn't just slow down fat synthesis. It also stimulates fatty acid oxidation. That's because malonyl-CoA normally inhibits the transfer of fatty acids into the mitochondria for oxidation. When malonyl-CoA levels drop, this inhibition is lifted, allowing fatty acids to be transported into the mitochondria, where they can be broken down for energy. So overall, this mechanism highlights how the cell tightly regulates energy production and storage, especially during energy scarcity, by switching from fatty acid synthesis to fatty acid oxidation. It's a fine example of the cell's adaptive strategies to maintain energy balance. Now, let's look at how the CAMP cascade functions when blood glucose levels are low. When glucagon or epinephrine are released, they activate this cascade, which is a key part of our body's response to fasting or low blood sugar. This activation leads to the phosphorylation of acetyl-CoA carboxylase through CAMP, dependent protein kinase, also known as pKa. Phosphorylating acetyl-CoA carboxylase inhibits its activity, and this is crucial because it shifts the metabolic balance. Instead of converting acetyl-CoA into fatty acids, the acetyl-CoA remains available for the synthesis of ketone bodies. And these ketone bodies become our alternative fuel source when glucose isn't readily available in the blood. So, this process essentially helps our body adapt by switching from glucose to ketone bodies, ensuring that our tissues, especially the brain, continue to get the energy they need during fasting. This ties back to the larger theme of how our metabolic pathways are finely tuned to maintain energy homeostasis under different physiological conditions. Now, let's take a closer look at the antagonistic effect of insulin. When blood glucose levels are high, insulin is released to help lower them. Interestingly, this effect is actually linked to the activation of an enzyme called protein phosphatase. What this means is that insulin, in this context, acts to activate protein phosphatase, which then plays a key role in reducing blood glucose levels. This process is a great example of how our body's regulatory mechanisms work in balance. It connects to the overall theme of how hormones like insulin help maintain homeostasis, ensuring our blood sugar stays within a healthy range. Now, let's look at how acetyl-CoA carboxylase is regulated by local metabolites. One key player here is malonyl-CoA, which is actually a product of fatty acid synthase. When malonyl-CoA levels are high, it promotes the inactive conformation of acetyl-CoA carboxylase. This means that the enzyme's activity decreases, leading to less production of malonyl-CoA. Since malonyl-CoA is the precursor for fatty acid synthesis, this effectively slows down the process when enough fatty acids have been produced. This is a classic example of feedback inhibition, where the product of a pathway helps regulate its own production. It ties into the overall theme of how metabolic pathways are tightly controlled to maintain balance within the cell. Now, let's talk about how citrate plays a crucial role in regulating fatty acid synthesis. When there's enough acetyl-CoA entering the Krebs cycle, citrate levels in the cell become high. This is a signal that energy and building blocks are plentiful. Citrate then allosterically activates acetyl-CoA carboxylase, or ACC. This enzyme is key because it commits acetyl-CoA to the process of fatty acid synthesis. Excess acetyl-CoA is converted into malonyl-CoA, which then serves as the building block for fatty acids. These fatty acids are ultimately stored for future energy needs. So, in essence, citrate acts as a metabolic switch, indicating when it's appropriate to turn on fatty acid production, tying back into our theme of metabolic regulation and energy storage. Now, let's look at how fatty acid synthesis from acetyl-CoA and malonyl-CoA occurs. This process involves a series of reactions that differ between bacteria and mammals. In bacteria, these reactions are catalyzed by 6 different enzymes, along with a separate acyl carrier protein, or ACP, which plays a crucial role in shuttling the growing fatty acid chain through the process. In contrast, mammals have a large multifunctional enzyme complex, known as fatty acid synthase, where each domain within this big polypeptide performs a specific step. Interestingly, this complex includes an ACP domain as well, but rather than being separate, it's integrated into the single polypeptide chain. The evolution of this mammalian enzyme appears to have involved gene fusion, meaning multiple genes that initially coded for separate proteins have merged into one continuous gene, resulting in this multifunctional enzyme. Another key point is the role of NADPH. It serves as the electron donor during the two critical reduction reactions in fatty acid synthesis. Without NADPH providing those electrons, these reduction steps wouldn't occur efficiently. Most of the NADPH used in these reactions is generated by the pentose phosphate pathway, which is a vital metabolic route, providing reducing power for biosynthetic processes like this one. So, in summary, fatty acid synthesis is a complex, evolutionarily adapted process that relies heavily on enzyme structure and cellular NADPH supply, all of which tie back into the broader themes of metabolic regulation and cellular energy management we've been discussing. Now let's take a closer look at fatty acid synthase and its prosthetic groups. These are crucial for its function. One key prosthetic group is the thiol on the side chain of a cysteine residue within the condensing enzyme domain. This thiol acts as a reactive site, allowing the enzyme to form a thioester bond with the growing fatty acid chain. Another important prosthetic group is the thiol of phosphopantethine. Structurally, it's quite similar to part of coenzyme A, which we've seen plays a central role in acyl group transfer. This similarity enables phosphopantethine to serve as a flexible arm, carrying the acyl intermediates between different active sites within the fatty acid synthase complex. Together, these prosthetic groups are essential because they facilitate the transfer and activation of substrates during fatty acid synthesis. They essentially act as molecular handoffs, ensuring the process runs smoothly and efficiently. Understanding these groups helps us appreciate how fatty acid synthase is a highly coordinated and complex enzyme system, integral to lipid metabolism. Let's take a closer look at phosphopantethine, or PANT for short. This molecule is attached covalently, meaning it's bonded securely, to the acyl carrier protein domain of fatty acid synthase through a phosphate ester linkage to a serine hydroxyl group. This attachment is crucial because it enables the transfer of fatty acid intermediates during synthesis. What's really fascinating is the structure of phosphopantethine itself. It has a long, flexible arm, which acts like a swinging tether. This flexibility allows the thiol group at the end of the arm to move freely from one active site to another within the enzyme complex. This mobility is essential for the function of fatty acid synthesis. It ensures that the reactive thiol can shuttle substrates efficiently between different catalytic centers, streamlining the entire process. So, in the context of our overall discussion on enzyme efficiency and complex coordination, phosphopantethine plays a key role by acting as a dynamic carrier, keeping the synthesis process smooth and coordinated. As each of the substrates, acetyl-CoA and malonyl-CoA, binds to the complex, the first step involves the oxygen atom of a serine hydroxyl group within the malonyl/acetyl-CoA transacylase enzyme domain. This oxygen acts as the attacking group in the reaction. What happens next is quite interesting. Each acetyl or malonyl group temporarily forms an ester linkage with this serine hydroxyl. This is a crucial step because it essentially holds the substrate in place temporarily. Then, the group is transferred from this ester linkage into a more stable thioester linkage with the phosphopantethine thiol of the acyl carrier protein, or ACP, domain. This transfer is vital for the subsequent steps in fatty acid synthesis. Finally, the acetate is transferred to a cysteine thiol group on the condensing enzyme domain, preparing it for the next phase in the process. This sequence of transfers and linkages is fundamental to how the complex orchestrates fatty acid chain elongation, connecting the initial substrate binding to the larger process of lipid biosynthesis. Now let's look at the condensation reaction, which is step 3 in this process. Essentially, this step involves the decarboxylation of the malonyl moiety. What this means is that a carbon dioxide molecule is removed from the malonyl group, making it more reactive. After this decarboxylation, a carbanion is formed. This is a negatively charged carbon species. This highly reactive carbanion then attacks the carbonyl carbon of the acetyl or acyl moiety. This attack is crucial because it forms a new carbon-carbon bond, extending the growing molecule. Understanding this step is key because it highlights how the enzyme facilitates the formation of longer chains, which is fundamental in processes like fatty acid synthesis. So, in essence, this step links the decarboxylation event with nucleophilic attack, enabling the chain to grow in a controlled, enzyme-assisted manner. Now let's look at this step in the process. The B ketone is reduced to an alcohol through an electron transfer from NADPH. Essentially, NADPH donates electrons, converting the ketone into an alcohol. Next, dehydration occurs, which removes a water molecule and results in the formation of a trans double bond. This step introduces a double bond with a specific geometric configuration, the trans form. Finally, this reduction by NADPH not only converts the ketone to an alcohol, but also leads to a saturated chain if further hydrogenation occurs. These steps are crucial because they modify the molecule's structure and properties, playing a key role in the overall metabolic pathway we're exploring. Now, after the growing fatty acid has been transferred from the phosphopantethene to the cysteine sulfhydryl group on the condensing enzyme, the cycle is ready to start again. This means that a new malonyl-CoA molecule will enter, and the process will repeat. Essentially, this step is crucial because it allows the fatty acid chain to keep elongating, one 2-carbon unit at a time. This cycle of transfer and addition is fundamental to how fatty acids are built in the cell. It highlights the efficiency and precision of the enzymatic process, ensuring continuous growth of the fatty acid chain throughout the synthesis. And this repetitive cycle is what underpins the overarching theme of how fatty acid synthesis is a highly coordinated and controlled process in our metabolism. When the fatty acid chain reaches 16 carbon atoms, something interesting happens. The thioesterase domain comes into play and catalyzes a hydrolysis reaction. This means it breaks the bond linking the fatty acid to the phosphopantethene group. Essentially, it releases the fatty acid from the complex. The final product in this process is a saturated fatty acid with 16 carbons, known as palmitate. This is significant because palmitate is the end product of the fatty acid synthase complex. So, in summary, once the fatty acid reaches this length, the thioesterase ensures it's released, completing the synthesis process. This step is crucial because it determines the length and the saturation of the fatty acid produced by the complex. Let's take a closer look at the overall process of fatty acid synthesis, focusing on palmitate. Ignoring the roles of H+ and water for simplicity, we can write a balanced equation for its synthesis, starting from acetyl-CoA. So, the key inputs include 8 molecules of acetyl-CoA, along with 14 molecules of NADPH and 7 ATP molecules. These are the energy and reducing power sources that drive the process. The outputs are palmitate itself, plus 14 NADP plus molecules, 8 CoA molecules, and the ADP and inorganic phosphate produced when ATP is used. Another way to look at this process is through the lens of malonate as an input. In that case, the reaction involves 1 acetyl-CoA and 7 malonyl-CoA molecules, along with the same 14 NADPH molecules. The net products are again palmitate, but this time with 7 molecules of CO2 released, along with the NADP+, CoA, ADP, and Pi. Now, it's important to understand where fatty acid synthesis occurs. It takes place in the cytosol, but interestingly, acetyl-CoA is generated inside the mitochondria. Since acetyl-CoA can't cross the mitochondrial membrane directly, it's transported to the cytosol via a shuttle system involving citrate. This shuttle mechanism is crucial because it links mitochondrial metabolism with cytosolic fatty acid synthesis, helping us see how different parts of the cell coordinate to produce fatty acids efficiently. Recognizing this connection is vital for understanding the overall regulation of lipid metabolism. Fatty acid synthase is transcriptionally regulated, which means its production is controlled at the gene expression level. In the liver, insulin plays a key role. It's a hormone released when blood glucose levels are high. When insulin is present, it stimulates the expression of fatty acid synthase, leading to the conversion of excess glucose into fat for storage. This process helps our body efficiently store energy for later use. The transcription factors that mediate insulin's stimulatory effect include USFs, or upstream stimulatory factors, and SREBP1, which stands for sterol response element binding protein 1. Now, it's interesting to note that SREBPs were initially identified for their role in regulating cholesterol synthesis, but they also influence fatty acid synthesis. Additionally, polyunsaturated fatty acids can actually diminish the transcription of the fatty acid synthase gene in liver cells. They do this by suppressing the production of SREBPs, which are essential for activating the gene. So overall, this regulation showcases how various factors, hormones like insulin and dietary components like polyunsaturated fats, interact to control fat synthesis in our body. Now, let's take a closer look at what happens inside fat cells. When fat cells are storing excess energy, they actually produce a hormone called leptin. Leptin plays a crucial role in regulating our body weight and fat metabolism. One of its main functions is to inhibit the expression of SREBP1 and fatty acid synthase, which are key players in fatty acid synthesis. Essentially, leptin signals the brain that there's enough fat stored, which helps to prevent further fat accumulation. It does this by decreasing our appetite, increasing how much energy we burn, and directly reducing the production of new fatty acids. This process is a great example of how our body maintains balance, especially in the context of overall energy and fat regulation, which ties into the broader themes we've been discussing about metabolic control. Elongation beyond the 16-carbon length of the palmitate product of fatty acid synthase mainly happens through enzymes associated with the endoplasmic reticulum, or ER. These ER enzymes are responsible for lengthening fatty acids that are produced by fatty acyl synthase, as well as those obtained from dietary polyunsaturated fatty acids. It's important to note that fatty acids need to be esterified to coenzyme to serve as suitable substrates for these elongation processes. When it comes to adding those extra carbons, malonyl-CoA acts as the donor of 2 carbon units. The process is quite similar to fatty acid synthesis, but with a key difference. Each step is catalyzed by different proteins rather than a single multifunctional enzyme. Now, the initial step of elongation, the condensation, is carried out by a family of enzymes called fatty acid elongases, or ELOVL enzymes. They catalyze this first critical step in building longer-chain fatty acids, especially very long chains. Connecting this all back to the bigger picture, these elongation processes are essential for producing the diverse array of fatty acids our bodies need, not just for energy storage, but also for membrane structure and signaling functions. So, desaturases are enzymes that introduce double bonds into fatty acid chains, but they do so at specific positions. This means that not all fatty acids can be modified freely. The enzymes target particular sites along the chain. In mammals, however, there's a limitation. Our cells can't produce double bonds at certain locations, specifically at the D12 position. That's a critical point because it means that if we want fatty acids with double bonds at those positions, we can't synthesize them ourselves. As a result, some polyunsaturated fatty acids become dietary essentials. One prime example is linoleic acid, which has 18 carbons and 2 cis double bonds at carbons 910 and 1213. Since our bodies can't make the double bond at D12, we must obtain linoleic acid from our diet to meet our nutritional needs. This highlights an important aspect of how our dietary intake directly influences our cell membrane composition and overall health, especially regarding essential fatty acids that our bodies can't produce on their own. When a double bond is formed in a fatty acid, it involves a series of specialized proteins located in the endoplasmic reticulum membrane. First, there's NADPH reductase, which is a flavoprotein that has Fad as its prosthetic group. This enzyme plays a crucial role in transferring electrons during the desaturation process. Next, we have cytochrome B5, which can either be a separate protein or a domain attached to the desaturase itself. Its main function is to facilitate electron transfer between NADPH reductase and the desaturase. Speaking of the desaturase, This enzyme is where the actual formation of the double bond occurs. It has an active site containing 2 iron atoms, which are complexed by histidine residues. These iron atoms are essential for catalyzing the desaturation reaction. Altogether, these proteins work in concert to introduce double bonds into fatty acids, a key step in producing unsaturated fats that are vital for cell membrane fluidity and function, tying into our broader discussion on lipid metabolism and cellular processes. The desaturase enzyme plays a crucial role in introducing double bonds into fatty acids through a process called a mixed-function oxidation reaction. Essentially, it involves the transfer of 4 electrons from oxygen, which gets reduced to 2 molecules of water, while the fatty acid is oxidized to form a double bond. This electron transfer is a stepwise process, starting with NADH, which donates electrons to the fat-containing reductase. From there, electrons pass through cytochrome B5 before reaching the desaturase enzyme itself. The order of this transfer, NADH to FAD, then cytochrome B5, and finally to the desaturase, is key to understanding how the reaction is tightly regulated. As the double bond forms in the fatty acid, 2 electrons are extracted, which completes the oxidation process. To put this into perspective, let's consider the desaturation of stearate, an 18-carbon saturated fatty acid, into oleate, an 18-carbon monounsaturated fatty acid. The overall reaction involves stearate combining with NADH, a proton, and oxygen, resulting in oleate, NADPH, and water. This process is fundamental because it modifies the fatty acid structure, impacting their properties and functions in biological systems. Connecting this to the overall theme, it highlights how enzymes like desaturases are essential for lipid metabolism and cellular function.